Namaskar dear students, welcome you to our channel by Gulab Sir. As a part of series of lectures, today's topic is T.w.engelmann's experiment on splitting of light to illuminate cladophora and know about the site of evolution of oxygen. It is very interesting, and he made it very interesting by having use of physics with biology. So it is just like a biophysics. Yes, all the sciences are integrated, only we need to have that discoveries and invention to connect all these sciences together to have some new invention and discoveries. One of such discovery made by T.w. Engelmann. He used alga cladophora and a prism to have or to know about which site of light or which type of light has more rate of photosynthesis and more evolution of oxygen. ऐसा कहा जाता है ना, जो न देखे रवि वो देखे कवि, और हमारे साइंस में तो मैं ये कह सकता हूँ, जो न देखे रवि और जो न देखे कवि वो सब कुछ देखे साइंटिस्ट. Although it is not a rhyme, but for knowledge and really unbelievable discoveries and invention, and therefore we salute to the scientists. Otherwise, if this experiment would have not there, we could have not come to know that really what is the role of red light in the process of photosynthesis. Now let us know that this is the experiment, this is the diagram to know about evolution of oxygen. You are always in confusion, and I know because I passed through that page that really how it could happen and how it happens, and therefore I was interesting. Definitely you are also interesting to know about. Now see, he is the scientist, one who used this prism. He used that suspension of aerobic bacteria and cladophora plant in a suspension medium. He allowed to pass that white light through this prism, and as it is already, we knew that light split into 7 colors, and I wrote here V-I-B-G-Y-O-R, Vibgyor. Now this light with that splitter, he allowed to pass through cladophora plus into suspension of bacteria. Bacteria are aerobic. Now when light was, or this cladophora was continuously illuminated for 1 to 2 hours, then he found that after some time, number of bacteria or quantity of bacteria found accumulated in the 2 regions, red region and blue region. There was either 1 or 2 or few bacteria were in other, but there were more quantity in red and blue region. Why? See the experiment. The imagination what he made to know about was really miraculous, and therefore I always salute to the scientist, one who made this type of discoveries. See, practically he proved that there is more rate of photosynthesis in the region of red and blue light, and therefore question asked in examination and we write one of the following type of light has highest absorption and action of that plants for production of photosynthesis. So to explain this action spectrum through chlorophyll, that he used this experiment and proved that there is a more rate of photosynthesis in the region of red light and blue light practically. And yes, therefore here I have drawn this absorption spectrum, action spectrum with chlorophyll B, chlorophyll A and carotenoid. See, this is a chlorophyll A which has more action, as chlorophyll A is a photocenter. It converts light energy to chemical energy. Rest of the other pigment other than chlorophyll A, chlorophyll B, C, D, bacteriochlorophyll, bacterioviridin, phycoerythrin, phycocyanin, phycobilin, xanthophyll, carotenoid, they are antenna pigment. They only absorb the light and pass on to the chlorophyll A. Therefore this blue light which showed, and yes, chlorophyll A is greenish blue in color, it has more excitement. That chlorophyll A molecule has more excitement. That is what magnesium present in the pore ring of chlorophyll A donate electron and carry out further process of photosynthesis, and that we learn in cyclic and non-cyclic photophosphorylation. Now see, here in the region of this blue color, this is a action spectrum, this 400 nanometer to 700 nanometer. this blue light and red light which lies between 680 to 700 nanometer, there is more rate of photosynthesis. How it is? It is practically the aerobic bacteria which require oxygen. Naturally, they will be in the region where there will be more oxygen. And yes, it is true that in the red light there was more oxygen and therefore bacteria could accumulate there. We know that when we will feel suffocated in one room, we will go to another room. Like this, same way that bacteria found oxygen in the region, red and blue light, therefore they accumulated there to have the oxygen for their livelihood. As well as they have more organic matter to have the food. And that is what proved. And I prepared these notes you go through. These are the notes, short note. Can be asked. Explain T.w.engelmann's experiment to show that there is a more evolution of oxygen in red and blue light, and you can use these notes. I hope this is very clear to you. You go through same thing what I explained, it is written over there, so need not to confuse, but you please practice this diagram to have the clear cut explanation if it is given in the examination. So this spectrum also you can use. This is a altogether explanation for this experiment along with absorption and exchange spectrum, very clearly mentioned there which color is for which chlorophyll pigments, as well as it is very clearly written that more number of bacteria absorbed in red or found observed in red light as well as blue light because of evolution of oxygen. Thus, Engelmann could prove that there is a more rate of photosynthesis in the region of red and blue light. And yes, because of this explanation, I am sure that if question asked in NEET about where there is a higher rate of action or higher rate of photosynthesis, definitely you will write blue and red. With this question and answer, I end up with this video. I am happy that you are liking our video and sharing and subscribing. So happy learning, enjoy learning.